Chlorophyll and hemoglobin, worlds apart, but very similar in structure. We'll discuss this on Revolution Against Evolution. Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Your co-host Rich Gear here as well. And Doug, I'm really kind of interested in this. I'm excited about it, but I really don't know a lot, except that the fact that I know hemoglobin is more in animal life and chlorophyll is more in plant life. Is that pretty good?
Yeah, and they have similar functions. Hemoglobin is in animals and is used in the blood to transport oxygen. But chlorophyll has a similar function in transporting carbon dioxide. And so the more carbon dioxide there is, the more plants, plants thrive on carbon dioxide. They fix it into their systems and create structures within the plant. And so they have different functions. And of course, when the animals eat the plants, they take in the chemistry of life from the plant and to use it to create the life within the body of the animal. And so it's a symbiosis between two very different types of life, but the structures of chlorophyll and hemoglobin are almost identical except for the central metal that it captures.
Yeah,
Human,
what is the central metal, Doug? You got hemoglobin is iron, right?
yeah, I'm going to show you
Okay, yeah.
I'm going to share with you the structure. Hemoglobin has iron in the center, whereas chlorophyll has magnesium in the center. And
Okay.
and this gives you a idea of what the differences are, but you can see that the structures of them are very similar. Chlorophyll has this long side chain on it, which
Yeah.
is part of the weak bonds that I talk about a lot in my book, Revolution Against Evolution, that there are structures that either like water or don't like water. And this long chain right here is sort of like an oil, and it doesn't like water. And so one of the things it does is it adheres to the cell membrane, and that's its purpose. These also have three-dimensional structures that you don't see here. So it's kind of the we have scientists who do what they can to figure this all out. And the more that they figure it out, the more amazed they should be. But a lot of the times they just get arrogant at the fact that they can understand this. And they figure that the more they understand, the easier it is for them to believe that it came about all by itself.
Well, I've said for years that oftentimes scientists, in their hubris, they have put a name on something, thinking that explains why and how it happened, how it came to be, and it doesn't do any such thing. But they kind of it's almost like burying their head in the sand, ignoring the fact of how complicated this is. I always and another thing we used to say, Doug, is if Darwin would have seen how complicated even something like a cell, a molecule here, a
Mm.
cell of hemoglobin or of chlorophyll, I don't think they would ever. Maybe they would have never come up with the evolutionary scenario. It would have been too daunting of a thing. But back in the day, 150 years ago, 200 years ago, they were able to come up with these ideas because things didn't seem quite so complex. But what you're saying, even though, and what you're also saying, there's a similarity, which is we'll get to that in a minute, but also how complicated at the same time. But the similarity between the plant and animal cells, but those similarities show what we would call a common designer, but the evolutionists would say it's a common origin, but it's so uniquely so different in the way I mean, even their very core, magnesium and iron, so different, that begs that question. But the similarity, so the plants can be absorbed by the animal life, they can be eaten and worked. It's like a symbiosis, isn't it?
Right, and the protein structures in the plants are utilized as nutrients for the animals that eat it. And so it all points to a creator God. And the fact that these porphyrin molecules that attach to the iron molecule in the middle of hemoglobin and the porphyrin molecules that attach to the magnesium in the chlorophyll, uh, the similarity of this speaks very loudly of a common designer. And without it, we wouldn't have life as we know it. Now, chlorophyll's principal role is the absorption of light, and predominantly in the blue and red portions of the spectrum. And it drives the photosynthetic process. So photosynthesis fixes the carbon dioxide in the atmosphere, and it uses the carbon dioxide to create structures in the of life that exist in the plant. Hemoglobin uses this same design with a iron molecule in the center to transport oxygen and to places where it's needed in the body. And so these are dynamic processes that react. There's a continuous reaction that goes on. So there's chemistry going on all the time that gives the life. So it's something that is irreversible. I mean, it just continues to run and run and run until death occurs.
What's fascinating, Doug, when you talk about, we've talked about this on many other shows, the hemoglobin molecule it synthesizes or processes oxygen, yet oxygen is a very destructive thing in its own self, right? I mean, oxygen prevents things from combining and developing, and yet without it, life cannot exist. It's this, which came first, the oxygen or the molecule? And well, they have to happen almost at the same time. It would seem like that they would have to the structure to absorb oxygen to work for life to work has to already be in place. It can't come in place in the same manner that it seemed like the carbon dioxide the plant would have to be ready to work. You mentioned earlier, this is going to sound like a little bit off, that one long chain to come out. And I talked about this, we talked about a little bit before the show started, but that it repels water, yet plants need water to live. I mean, don't they? I mean, it's really strange. All
Well,
these kind of,
the repelling of water is part of the function of the binding process in
yeah.
plants, and there's parts of the chlorophyll that attract water, the ones that
Oh,
have
okay.
oxygen in it, and so they, and then there's ionic bonds and covalent bonds, and these are all things that contribute to the structure in the three-dimensional shape of it that creates the reaction. But
Now, is that three-dimensional structure? I'm assuming that's critical to the function of the molecule as well. Am I correct? The
that's
three-dimensional
correct, yes.
shape of it. So, well,
So
you want to talk a little bit about that?
you can make chlorophyll in the lab or hemoglobin in the lab. That's a something that I haven't actually tracked down. I think so. There's somebody that actually has done that, but it's something that would take years of planning and a lot of different types of uh, intelligence to do. Now, you can't
Yeah.
expect a reaction of that sort to occur in just a warm pond someplace of chemicals that were created by zapping uh, hydrogen and ammonia and nitrogen gas and things like that to create uh, a soup of prebiotic soup that we talk about. And
Yeah.
I in this little article I sent you, Rich, there is a
Yeah.
list of things that happen when they make chlorophyll. Well, what in not in the lab, but the way that life makes it. And it's a multi-step process. It's broken down in several major phases. The first one is the formation of a glutamyl to transfer RNA.
Okay. I
Now,
gotcha.
the transfer RNA is like part of the DNA, uh, and the DNA sort of spins off a a chain of a gene, and it creates the transfer RNA. The transfer RNA is where it codes for it attaches to amino acids and it transfers amino acids to a protein. The pathway becomes with amino acid glutamate, which is ligated to a transfer RNA molecule by glutamyl transfer RNA synthetase, forming glutamyl transfer RNA glutamine. This serves as an initial substrate for the synthesis of the 5-amino valinic acid, the universal precursor of all tetrapyrrole compounds, including chlorophyll and heme. Now, I'm having trouble pronouncing all this.
Yeah, I don't blame you. I tell you, we get done with this, Doug, the eyes are going to glaze over, but you're going to see how complicated this really is, aren't you?
That's all. That's the purpose of this little exercise. Is to
Yeah,
say, well, this is
my brain
all
is
great.
already kind of
And
swirling.
then this is the first thing where and I sort of understand what it's saying is that you got this transfer RNA, which is this little mechanism that hangs onto a glutamine, and it's ready to do the next step. And then you have the synthesis of the 5-amino valinic acid, the two-step reaction catalyzed by the glutamyl transfer RNase reductase and glutamate I-semialdehyde aminotransferase that converts glutamyl transfer RNA to alanine. This is the key regulated step responsive to light and developmental cues. Poor for bilirubin formation. And then you have two molecules of alanine are condensed by alanine dehydrase to form porphobilinogen, a monopyrrole. So a monopyrrole would be one of these um, hexagon molecules that form in the uh, in the that there's four of them that connect together to hold the iron or the magnesium in place.
Oh, I see. Okay.
And then formation of uroporphyrinyl 3. Four molecules of porphobilinogen are assembled as the first cyclic tetrapyrrole. Uroporphyrinyl 3 via porphyrin and uroporphyrinyl 3 synthetase.
Uroporphyrinogen. Wow.
And
They don't make anything short, do they, Doug?
no, if you break the structure of those words down, you can sort of understand what it
Yeah.
is if you know what those little components are. We have another step going on here, which I'm not going to explain, and then you have insertion of the magnesium. And that's this critical juncture enzyme magnesium chelatase inserts a magnesium ion into the in the porphyrin and distinguishing chlorophyll biosynthesis from the synthesis of the heme, which incorporates iron instead. So you have
Okay. All right. I
so
got
it's
that.
actually showing that the mechanisms to make both of these molecules are related.
Very similar. Okay. Yeah.
And through several methylation, cyclization, and reduction steps, pathway proceeds from magnesium protoporphyrin the 9 through protochlorophyllide, a direct precursor to chlorophyllide. Then there's a
Yeah.
reduction that takes place. Another step that goes on is there's a strictly regulated and is bottleneck for greening onto light exposure and the formation of chlorophyll A and B. So there's two different types of chlorophyll.
Okay. Oh, I didn't know that.
And so, and then it's esterified with a phytol chain forming chlorophyll A. So if you want to really know this and to figure it out and to understand it, you can go into your artificial intelligence generators that you have on this and type in chlorophyll synthesis or hemoglobin synthesis. And so both of these are essential to life, but they are in far-reaching ends of the spectrum of where evolution would want you to believe that they occurred from. But these would tie you together to a common creator, not a common ancestor.
We talked a little bit about the symbiosis, like the animals need they can eat the plants and they absorb the nutrients. But we didn't talk about, and to me, it's fascinating, at least from a layman's perspective, that plants generate carbon dioxide as well as the animals generate oxygen, which each side needs the other. Am I correct in that? Right? The more plants, the more oxygen, which is good for animal life. Then the animal life will come up and then produces carbon dioxide, which is good for plant life. Am I right there or not?
Right, and I think that people don't understand that the carbon dioxide is an essential, is not a poison. It is a essential part of life, and the more that you have, the more lush you're going to have your forests, because that's what they feed upon.
I know, and I've never
Right
understood this.
to, and
Go ahead.
there's a plant in Norway, I guess, where they're taking carbon dioxide out of the atmosphere and trying to put it underground and keep it away from the atmosphere. You know, they're trying to get rid of excess carbon dioxide and get it out of the environment, and that's total foolishness. Now
Yeah,
they confuse it with carbon monoxide. The
yeah.
carbon monoxide is a poison, but that's something that is easily broken down in the atmosphere, and it turns back into carbon dioxide. So it's it's kind of a strange world we live in, where the things that are essential to life are called poisons, and I really am
Well,
baffled.
Doug, it kind of reminds me, even this is a scientific thing. We find that even in the moral and the spiritual stuff, what's good is now labeled bad, and what's bad is labeled good. And it's really strange. You are correct, Doug. The world we live in is strange, but it even affects the physical. You think something as immutable as evidential scientific things would be what they are, but they've made, like you say, the carbon dioxide, which is essential for life, an evil thing. It doesn't make, I mean, it makes no, I've never understood this when they talked about this for a long time. I mean, you can always talk about, Doug, if you have too much of something, it can be bad for you. But Doug, you've talked about you have too much water, you get too much water and guess what happened? You drown, you know?
Right, yeah.
I mean, anything too much of, you're going to die. I remember years ago, Doug, this is something a little bit different, but when people got really down on cyanide in vitamin B17 or something, the Laetrile, this was way back in the day, and you said, yeah, but our bodies need a little bit of that. It helps the cellulars turn on and off. And I looked at the B vitamins, and they got it's not cyanide per se, but it's a product of cyanide in it. It seems to me when I was reading the ingredients of some of the B vitamins, and I'm probably going way off the thing there, but it just seems like so many things that really in, in small doses or whatever, or the proper things are actually good for you. And they try to blanket it and make it all horrible. Yet they'll swallow all kinds of other things, which are totally terrible for you. And they'll say that's good. They'll take that and make that as good. I don't understand it at all.
Yeah, something else I found out while researching this was that there's another compound called cytochrome C. Have you heard of that?
I have not.
Okay, cytochrome C is something that is a very similar structure as chlorophyll and hemoglobin, but it's used in the mitochondria and it transports electrons. Uh,
Wow.
yeah, and
This is
it's
both in animal and plant life, Doug? Is
yeah,
this
in
same?
animal and plant life, and in the
Oh,
mitochondria, it's actually used in the energy production in the ATP synthesis.
ATP, okay.
So, Cookie and I are starting to take some ATP and getting all energetic, right, Cookie?
Right.
But that's a very wonderful process that occurs in the body, and it's actually a motor that rotates when it's producing the ATP.
Yeah, I've done some drawings for Jerry Bergman on that, the ATP. It's a microscopic motor. It's amazing. ATP, yeah. But I don't remember that other compound or what, or that what'd you call it, Doug? What was the name of it? What'd you call it?
Adenosine triphosphate is what it's called.
Oh yeah, that I've heard of. Yeah, but using ATP anyway.
That's what ATP stands for.
That's what that is. But what's the other thing you talked about just before that? It was using mitochondria.
Oh, cytochrome C
Yeah,
is
cytochrome C.
part of the
Yeah,
process, and it
right.
actually carries a I think it's an electron.
Is
Uh,
that what helps the motor run, Doug?
I think so, possibly, yeah.
Okay. All right. I don't know. So yeah, it's interesting. All right, but it's used in both hemoglobin, or I mean, animal and plant life, that particular thing. But it's
Yeah.
but it's similar. Okay. Is the structure similar, Doug?
Yes, it's quite similar to chlorophyll and hemoglobin.
Hemoglobin. Okay. Cool.
So, I'm looking here at the synthesis of hemoglobin, formation of delta aminovalinic acid. Okay, that's similar to what is done in chlorophyll. Okay, so the synthesis in the animal actually follows quite a bit uh, the same process steps that it ends up with chlorophyll. And so
Yeah.
so
Until the insertion of
I was
iron.
looking at this and I says this is all very similar except the finally enzyme ferrochelatase incorporates a ferrous ion, which is iron, into the protoporphyrin to yield the heme group. Okay, so the hemoglobin.
No, but it's interesting because I noticed that this, like, it's step 9 in the insertion of iron, where it's step 6 in the chlorophyll. And yet there's a
Right.
lot more going on in the chlorophyll after that. There's only like one major step after the insertion of the iron. There's 3 steps after the insertion of the magnesium. So it's kind of interesting how that it's similar, but there's wildly different things that are going on here. But again, I think your point that that it reveals a common designer, that this is good, So we got a few minutes left, Doug. How do you want to finish this up?
Well, one of the things is this little treatise here talks about mutations that take place in the synthesis of hemoglobin, and there's a one of them is called thalassemia. The other one is called sickle cell disease.
Oh, so these are
And
these are
these
detrimental
are these
mutations.
are point mutations that cause that in the formation of hemoglobin that cause the three-dimensional structure to be altered from
Mhm.
what it actually should be, and it causes anemia in both of these cases. And so we understand that is point mutations are bad, not good. In evolution, mutations are what drives the changes
Right.
in genetic structures. Whereas we understand in the beginning, when God created life, He created all these different varieties ex nihilo. He created it out of His own mind, and He created it with a purpose. But we do have changes that take place with epigenetics, where you have things that are turned on or turned off in the genetic structure based upon environment. And that's all something that was designed as well. And for people to not recognize that is a mystery to me why people don't, who are biologists or chemists or or geneticists, why they don't recognize this and they should be first in church every day.
You think, yeah, yeah. Well, I think that they conflate the idea of adding something new to changing something that was already there. The environment can do that, but it can't add anything really new. And anything that changes the genome, it's generally it's at best neutral, but most of the time it's negative. So we still have the same thing, and that's understood from a creationary viewpoint that everything was created perfect, like you said, but there was a corruption and there was a death inserted in, and that's where we live today, right, Doug?
Mhm. So, one last point here is that there is recent research that's going on by the use of gene editing to restore some of these iron deficiencies and the
Hmm.
mutations that we're talking about by using the gene editing. And they're hoping to create cures for sickle cell disease, thalassemia, and iron deficiency, and porphyrias. These are rare disorders of the enzyme deficiencies um, in the synthetic pathway of the hemoglobin. And so we look at these things as possibilities. We can use the technology to understand what it should be, what the original created genetic code would have been, and we can cure some of these diseases.
That's a good
Well,
thing.
this is the possibilities that we can look forward to. We'll see you next time on Revolution Against Evolution.